Greetings. Greetings. Hello. So my name is Marki, and I am an instructor in the laboratory of Klaus Ley at the Jolla Institute. And thank you very much for this opportunity. I'm delighted to share with you our recent work, our recent results about the discovery of elongated neutrophil-derived structures. But I will tell you a little bit more than just these structures, because the way to discovery of these structures led us through neutrophil rolling and tethers of neutrophils, and I would like to give you a little introduction of those things as well. So neutrophils are the most abundant cells in the human blood, most abundant immune cells in the human blood, and their primary role is protecting us from infections and bacterial and fungal infections. And they also have a role in tissue remodeling. So neutrophils are made in the bone marrow, and from there they are released into the blood circulation. And with the blood they travel to all sites in the human body. And if they encounter an inflamed vessel wall, then they begin to extravasate into that tissue. And this extravasation cascade is a multi-step cascade that entails the rolling of the neutrophils on the vessel wall. And this is what the movie shows you as well here, Intravital recording. And then arrest and crawling on the vessel wall and transmigration into the extravascular space. This is the last picture here, last section of this cartoon. So in the Ley lab, my work focuses on the rolling part of this cascade. And one of the very exciting questions for us is that how can the neutrophils maintain rolling under these tremendous forces exerted by the bloodstream? So rolling neutrophils velocity is about 1,000 times less compared to the blood flow velocity. So they really need to cling on to the vessel wall to survive that. And well, one of the mechanisms that enables neutrophils in it is called the tether formation. So tethers are long, thin membrane structures pulled out from the adhered microvilli of rolling neutrophils. On this movie here, you see mouse neutrophil, bone marrow-derived neutrophil that we labeled for plasma membrane. This is the green channel, and this is a side view flow chamber microfluidic device that we designed specifically for the purpose of visualizing the tether formation from a lateral perspective of the rolling cell. So what you can see here is that as the cell rolls by, these long, thin structures form behind it. And here on these still images, you can observe these structures with a higher signal-to-noise ratio. You can see that neutrophil can form multiple of these tethers at the same time, and with multiple lengths as well. Now, there are actually quite a few in vitro studies out there which looked at these structures in different techniques. And in vitro, it is well established that tethers are actually load-bearing structures. So, however, it was until we did some intravital work, it was not known whether this is true in vivo as well or not. And in the next slide, I would like to show you a little bit about this in vivo work that we have done into the direction. So to interrogate whether tethers form consistently in vivo as well, and whether they are load-bearing in vivo or not, we turn to the cremaster intravital microscopy model. So in this model, we prepared out the cremaster muscle of anesthetized mice. And these muscles are very thin and highly transparent, so they offer a great opportunity for high detail imaging. And we injected these mice with antibody against the neutrophil surface. That antibody was against the Ly6G surface antigen. And this is what the magenta color shows you here. And so we have seen multiple examples of tether formation of rolling neutrophils. Like here you see 3 columns. Each column shows you like consecutive frames of the same cell rolling from left to the right. and you see this thin structure behind the cell around the third frame, or maybe even the second frame and the third column. These are the tethers that we have observed. And to investigate or to test whether these tethers are load-bearing, what we did is we measured the rolling neutrophil displacement right before and right after the disappearance of the tether. So right after the tether disappearance, the neutrophil rolled a little bit longer during that frame time, indicating that when the tether fails, the neutrophil makes a jump, makes a short acceleration. And as you see here on this plotting, in most cases the displacement right after tether failure was longer compared to right before tether failure. So most neutrophils make a jump after their tether failure. This suggests that the tethers are load-bearing structures in vivo as well. So next, during these experiments, we have observed many elongated structures on the vessel wall. And these elongated structures were lysis G positive, which shows that they are derived from neutrophils. They're magenta positive, so lysis G positive. And what we observed is that some of these elongated structures are stuck to the vessel wall, others were sliding along the vessel wall, and yet others were circulating with the bloodstream. So when we took blood from these mice, we were able to detect in the blood plasma these elongated structures as well. We named these structures as elongated neutrophil-derived structures, or shortly ENDS. So this name entails that these structures are elongated and neutrophil-derived. Well, of course, we were wondering how do these structures form, and from our perspective, the most plausible explanation was that these form when neutrophils lose their tethers. So, and indeed, we could find some examples for that. In this movie, in this montage, you see neutrophils rolling from the left to the right, and as they roll, some of them leave behind tethers, or elongated structures on the vessel wall. And those ends are indicated with the yellow rectangle. And as these neutrophils formed ends, actually they showed unstable rolling velocity, unstable rolling as well. This is what you can see here now on this graph. So the full circles indicate the rolling velocity of ends forming neutrophils, and their speed was always higher than of the velocity of the non-ends forming cells. So from these observations, we concluded two things. One is that the tethers are indeed load-bearing, and tether failure will result in unstable rolling, just as expected. The second thing is that since in our movies we have seen only about 6 or 7% neutrophils showing tether failure, we thought that this might be connected to some subset of the neutrophils. So we didn't follow up this question yet, but this will be very interesting to find out whether this is true or not, and which subsets are prone to tether failure. So far I have shown to you results measured on mouse neutrophils. And to link this to human setting, we have turned to flow chamber experiments on human neutrophils isolated from healthy human donor's blood. We labeled the neutrophil surface with a CD16 antibody, and these antibodies, they label very homogeneously and brightly the neutrophil surface, and we expose them to stepwise increasing shear stress. And as you can see, as the shear stress becomes higher and higher, or the flow increases, neutrophils start to form more and more tethers, and they start to fall off from the vessel wall, or from the flow chamber wall, and they leave behind these elongated structures. To analyze this, to quantitatively analyze these records, we have developed image segmentation method with Imaris that allowed us to specifically gate on the cell body or the tethers and or the ends in these records. And what our analysis showed of these movies is that, as expected, with increasing perfusion rate, the cells showed increasing rolling speed. And on E-selectin substrate, the rolling was more stable than on the P-selectin. And with increasing shear stress, more and more cells fall off from the vessel wall. And additionally, so the most relevant to this topic here is actually that with increasing shear stress, the neutrophils formed more and more ends as well. So we quantify this by measuring the total area of the yellow signal on these records. So from this we can conclude that human neutrophils can do the same thing, even though we have in vitro evidence for that only. And that ENDS formation is related to the shear stress level. So I mentioned to you earlier that the elongated structures were also detectable in the blood plasma. And this raises the question, so how do these structures get into the blood? Since they form on the vessel wall, somehow probably they detach from the vessel wall, or what exactly happens there? So what we have observed as well, that the ends forming in the ends, the sliding ends often detached and went into the circulation. So on the upper movie here, you can see a venule, a small vein of a mouse, and the yellow rectangle shows you how an end slides on the vessel wall and then suddenly disappears, suggesting that it detached from the vessel wall at that point. So it looks like when ends form, they can slide along the vessel wall and then they can suddenly detach from the vessel and go into the circulation. So to model this in a flow chamber, what we did is we prepared the flow chamber which was half coated with selectins, and half of it was coated with selectins, the other half was not coated with selectin. And in these half-coated chambers, we could recapitulate this observation that sliding ends, they detach when they reach a certain point where there are not enough adhesion molecules on the wall. So in the microcirculation, it is known that selectins are mainly expressed in the venules, small veins, and their expression density decreases as we proceed further toward the larger veins. So an explanation for ends getting into the circulation could be is that as they slide these ends, they reach a level where there are not that many selectins anymore, and they just fly away with the flow. They cannot hold on anymore there. So to summarize these things that I showed to you so far, here are three pictograms. The first one shows you that rolling neutrophils, they form tethers, and the second one shows you that these tethers break off from the rolling neutrophils, and the third one shows you that the remaining ends, they either stick or slide on the vessel wall or circulate with the blood flow. So next, we thought that these ends, these are basically a new category of particles by the neutrophils. And to put them into context, we turned at the literature, we looked around what are the secretory mechanisms of neutrophils, and there actually are at least four such secretory mechanisms or major secretory mechanisms described out there. So as the neutrophils get closer and closer to the inflamed tissue, they get more and more activated, and they release more and more things into their environment. And they do that by extracellular vesicle release, which these are the exosomes and ectosomes and apoptotic bodies. Then they do that by DNA release. This could be mitochondrial or nuclear DNA release, or NET formation, which is standing for neutrophil extracellular trap formation. And neutrophils also release a lot of substance through degranulation, and migrating neutrophils can also leave behind trails as they go through the tissue or go along the vessel wall. And so we think that the ENDS is in addition to this group, to this secretory mechanism group, and ENDS release is actually connected to rolling of the neutrophils. So next we aim to characterize a little better these ENDS, and so we performed a correlative electron microscopy experiment, which confirmed that this is an elongated structure. And we also did storm imaging, which confirmed that the diameter of these structures is about 115 nanometer. Next, we did some microscopy experiments to find out whether ENDS contain any organelles, and we found only cytoplasm inside, no ER, no mitochondria, no nucleus or DNA. Then we set up an assay for proteomics analysis. So for this, we optimized a flow chamber experiment for producing as much as possible ENDS without neutrophil contamination. And for this, we applied a couple of tricks. So we have rolled the neutrophils in these chambers in the absence of calcium and magnesium to avoid arrest of these cells. On the other hand, we increased the P-selectin coating in these chambers to allow the cells maintain effective rolling under these low calcium, low magnesium conditions. Then, after producing these ENDS, production of ENDS, we washed through the chamber at high flow to get rid of every possible cell which could cling on somewhere. And after that, we harvested the ENDS with SDS buffer. And then we run LC-MS proteomics on these samples. So the results showed us that ENDS are enriched for a couple of proteins, like 57 or so proteins compared to the whole neutrophils. And but here I would like to focus your attention on the S100, A8, and A9 proteins, which are known inflammatory mediators. These are damage-associated proteins in humans and also in mice. So next, we wanted to see whether the ENDS could be a source of protein S100, A8, and A9 in the circulation, or in an in vitro experiment like this one here. So we produced ENDS in a flow chamber, and we imaged these structures over 4 hours to see how the shape changes, whether they degrade or not. And we found that they indeed degrade, they round up, and they become shorter as well. This is what the graph shows to you here. So the red curve indicates the sphericity of the ENDS, and you see that it increases over time, and you see the END size with the black curve that decreases over time. So from this, we concluded that ENDS, they do round up with time or degrade. And we measured in the supernatant of these chambers with an ELISA kit the S100, A8, A9 complex amount, and we found that with time the quantity increases. So with time, these structures release more and more and more S100, A8, and A9. So to add this information to our summary slide here, we added a new pictogram that indicates with time ENDS, they degrade and they release S100, A8, A9, and possibly other mediators as well into the circulation. This is symbolized here with these yellow dots. So to find out the relevance of this to human health and disease, or to put this into a human context, we turn to septic samples, and we asked the question whether we can detect ENDS in blood of septic patients or not. So we wanted to test whether this is true or not in a human disease context as well. And for this, we decided to analyze blood of septic patients. So for this type of, for a quantitative screening method, we developed an image stream or imaging cytometry-based scanning approach, and in collaboration with Yoav Altman at Sanford Burnham. And so why this instrument was ideal for us is because this instrument gives information about the shape of the microparticle as well. So how this instrument works? Well, imagine this is a flow cytometer, but instead of one data point for each event, you have one picture for each event. And you can run all sorts of image analysis on that picture, and from that you can derive the shape of the object. So I'd like to show you here some technical details about this scanning. So first, the labeling approach. Well, we generally use a double labeling approach, and the goal of that was to label the ENDS with two different color antibodies, so that we would be able to distinguish between antibody clumps and between real objects. So we took only those events, real neutrophil-derived, which were positive for both color antibodies, like Ly6G-FIT-C and Ly6G647, or in case of humans, CD66B-FIT-C and CD16-647. So the goal of this was essentially to analyze the shape of these particles. And for that, the gating strategy is shown here under the third point. So with this method, we were able to detect significantly increased amount of ENDS in blood of mice injected with LPS. So before injection, the mice had almost no ENDS in their blood, and 2 hours after LPS injection, they had significantly increased amount, about 600, 700 ENDS per microliter of blood plasma. So for the human scenario, we collected blood plasma from septic patients that were diagnosed based on 2 or more search criteria and suspected infection. This was a very heterogeneous population. As you see, they had all kind of primary and secondary diseases. While this is a general problem for sepsis research for the field, this is a very heterogeneous disease group or patient group, and it requires a lot of effort to stratify it properly. The results for the septic samples looks the following. So here you can see a raw data that was generated by the image stream instrument. And the upper block here, you can observe the spherical objects. So all of these objects are double positive for CD66B and CD16, which gives us assurance that they indeed come from neutrophils and they are not antibody clumps. So in the amongst the spherical objects, we could observe examples for double positives for annexin and tetraspanins, and also double negative for both, or one negative, one positive for each one of these. On the other hand, we also detected multiple examples for elongated objects or ENDS, and this is what you can see in the lower block here. And these ENDS were all negative for tetraspanin and annexin 5. So we were interested in the shape of these objects. That's what we use to call an object ENDS or a spherical object. And to do that, we measured the aspect ratio of these events. The aspect ratio is the ratio between the height and the width of that structure. And so if something is a perfect sphere, then it will have aspect ratio 1. And if it would be a perfect line, then it would be 0, or very, very close to 0. So as you see, the shape of these objects changed along a continuum, And so we had to pick somewhere a cutoff, and that cutoff we chose at 0.6 aspect ratio. So all the objects that had aspect ratio less than 0.6 were elongated for us, and we call them ENDS. And those which had aspect ratio larger than 0.6 were spherical, and we call them. So we think that they might also entail the fragmented or degraded ENDS as well. So on this graph, you can see septic and healthy data. The septic one is indicated with gray symbols, and the septic is with the gray symbols, and healthy with the black symbols. And we compare the total END count in healthy and septic conditions. You can see that there is a highly significant difference between the two. Additionally, we measure the S100A8A9 content of blood plasma in healthy and septic patients, and what we saw is that in the septic patients, there was a correlation between the S100A8A9 content and the number of fragmented ends. So the fragmented ends, they are spherical shaped, right? And they have aspect ratio larger than 0.6. And our in vitro experiments indicated that in the supernatant of fragmented ends, more and more S100A8A9 appears with time. So, which looks back to our in vitro imaging and suggests that END degradation happens in humans as well, in the blood, and it will result in increased S100A9 content of blood plasma. So this is what we published in our recent publication with the mouse and human evidences together. And from here on, I would like to show you some data that is not published yet. So when looking at this cascade, multiple questions could arise, and I just picked two questions here that I would like to further discuss with you. First one, what could be the molecular mechanism of tether failure? And the second, what happens to the ends in the circulation? How do they get cleared? So to address the first question, I would like to show you a little bit on the connection between the plasma membrane and the F-actin cortex of eukaryotic cells. So it was established already about 20 years ago that phosphoinositides can anchor together or have an important role in anchoring together the plasma membrane with the F-actin cortex. And there are these anchoring proteins which can bind phosphoinositides. These are these yellow structures in the plasma membrane and the F-actin. And it was also discovered that upon stimulation of phospholipase C, for example, through inflammatory mediators, this bridge can be broken up by cleavage of the phosphoinositides. And so when this axis breaks up, the plasma membrane becomes easily extractable, and the cell will lose membrane from itself. So to test whether this mechanism could have a role in tether failure, we set up some inhibitor experiments. So we exposed the cells to phenylarsine oxide and wortmannin to block its PI4 kinase activity and decrease the phosphoinositide phosphate content in the plasma membrane. And we also exposed the cells to latronculine and also inomycin. And on the next slide, I would like to show you the results of this. So here you can see mouse neutrophils rolling in a flow chamber. And these neutrophils were either untreated or were treated with latronculine, phenylarsine oxide, or wortmannin inhibitors. And as you can see, The control cells under these conditions did not form ends. They didn't leave behind ends. On the other hand, the Lachonkulin, Pao, and Wortmanin, they did a lot of ends formation. So this to further test this the relevance of this mechanism in tether failure, we set up an experiment where we could expose the neutrophils to ionomycin, rolling neutrophils to ionomycin. So this is a little tricky experiment because ionomycin, which is an ionophore, and it induces the streaming of calcium into the intracellular space. It activates the integrins of the neutrophils and makes them arrest suddenly. So to come around this problem, we have turned to HL60 cells, undifferentiated HL60 cells, which don't have complete integrin machinery, and therefore they cannot be arrested by ionomycin. Furthermore, we transfected these cells with the PIP2 biosensor PH-PLC RFD. And we roll these cells on a selectin substrate, and we superfuse them with ionomycin. And what we observed is that when the ionomycin reached the cells, these HL60 cells started to form many, many long tethers. The PIP2 biosensor detached from the plasma membrane and spread all over the cytoplasm. And eventually these tethers, they broke off from the cell and they became ends. This further provides evidence that the phosphoinositides F-actin bridge or axis holds together the cell, and it has an important role in tether failure. So based on that, we have further candidates in mind to dissect further what could be the molecular mechanism behind that. Here you can see a brief list of these candidates: bar proteins, ERM proteins, or myosins. So next topic that I would like to touch on briefly is that what happens to the ends in the circulation. Well, to address this question, first we did an experiment where we injected the mice with LPS to induce endotoxemia, and then we bled these mice multiple times following the injection. And we measured in each sample the end count, and what we found is that the end count, they drop after 4 hours and 8 hours in the blood, suggesting that somehow ends disappear or they degrade or they are removed from the circulation by some sort of mechanism. Well, to further investigate this, we have set up some mesentery imaging experiments. So for this, we have designed a new mouse preparation stage, and with the help of that stage, we imaged the mesentery circulation following LPS injection. So without LPS injection, we saw many neutrophils rolling on the vessel wall here. This is what you see here on the left movie. And however, 1.5 hours after LPS injection, we couldn't see any ends on the vessel wall, and we saw just a few neutrophils rolling, and we saw mostly cytoplasts actually along the vessel wall. So, but you know, the bottom line is that the ends were not visible in this vascular bed, even though we injected the LPS right there. So, where did the ends go from here? Where did they disappear within 1.5 hours? So next, we injected we set up another experiment where where our goal was to image the cremaster muscle through 1.5 hours following LPS injection. And this experiment showed us that after LPS injection, even though neutrophil rolling is not apparent in this vascular bed, the ends, they just start to appear on the vessel wall without apparent neutrophil rolling. Here you can see some examples of this. So about 1 hour after LPS injection, the movie shows there are some elongated structures along the microvessels of the cremaster muscle. And here you can see some snapshots of these elongated structures. So from this suggests that following LPS injection into the intraperitoneal space, the ends, they probably form in that vascular bed, and then they travel around with the circulation, and they stick to the cremaster microvasculature. So ends might populate other organs than their organ of origin. Like this, they might contribute to remote organ injury. However, these are preliminary results, and of course it requires further investigation. So this suggests that ends could travel and stick to all sorts of vessel beds in the inflamed body. And here I would like to show you one more data regarding what can happen to the ends. So we found that monocytes and macrophages can phagocytose them in a flow chamber. On this movie here, you see how a human monocyte picks up human ends in a flow chamber. As you see, over 3 hours, it is cleared up very well. And on the next movie, you can see how a mouse macrophage cleans up mouse ends, again in a flow chamber experiment. So this suggests that these ends, they could be cleared from the circulation by monocytes and macrophages. So to complete these pictograms, this summary slide, we added here two drawings. One shows that ends, they can populate remote organs. They can stick to the vessel after prolonged circulation, probably. And the last one here shows that ends can be phagocytosed by monocytes and macrophages. So with this, I would like to thank your attention. And these are the great people with whom we did this work. And thank you very much.